Welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding mechanism of transcription from the molecular basis of inheritance in detail. So here what is transcription or how we can define the term known as transcription. Transcription, it can be defined as the mechanism or it is the process through which transferring of genetic information it takes place from DNA to RNA, and here it is termed as what? Transcription. And here the mechanism of transcription it is observed both in case of prokaryotes and even in case of eukaryotes. Is it clear? So let me write it on the board. Transcription can be defined as the mechanism or process through which transferring of genetic information from DNA to RNA takes place. From DNA to RNA takes place. So here it is termed as what? Transcription. And here the mechanism of transcription it is observed both in case of prokaryotes and even in case of eukaryotes. So let me write it here. Transcription is observed both in prokaryotes and even in case of eukaryotes. So here likewise the replication mechanism, even in case of the transcription mechanism, the complementarity relationship it is applicable. Is it clear? So let me write it here. Likewise, the replication mechanism even in transcription complementarity relationship is applicable. Is it clear? So here though the specific mechanism of transcription it remains very similar with the mechanism of replication, but still there are basic differences in between replication and as well as in case of transcription. So here in case of replication, the entire double-stranded DNA it undergoes duplication, but whereas in case of transcription only a certain segment of DNA it undergoes duplication in order to yield RNA. Is it clear? So let me write it here. In replication, the entire double-stranded DNA undergoes duplication to yield daughter DNA. But whereas in case of transcription, only certain segment of DNA undergoes duplication. And in addition to this, In between replication and as well as transcription, there are some more other additional differences here. So here in case of replication, in replication, adenine base pairs with thymine. Adenine base pairs with thymine. But whereas in transcription, adenine base pairs with uracil. And in addition to this, in case of replication, both the single stranded DNAs which proceeds in 3' to 5' polarity and as well as 5' to 3' polarity, they act as templates in order to synthesize the DNA in 5' to 3' polarity. Is it clear? In case of the replication mechanism, both the single stranded DNAs which proceeds in 5' to 3' polarity and as well as 3' to 5' polarity, they act as templates in order to synthesize the DNA in 5' to 3' polarity. But whereas in transcription, only the specific DNA strand which proceeds in 3' to 5' polarity, it acts as template in order to synthesize the RNA during the transcription mechanism. Is it clear? So let me write it here. In replication, both the DNA strands which proceeds in 3' to 5' polarity, and 5' to 3' polarity act as templates in order to synthesize daughter DNA. But whereas in transcription, only the specific DNA strand, which remains in 3' to 5' polarity, act as template in order to synthesize RNA in 5' to 3' polarity. So here you make it a small clarification. Why in case of transcription, both the DNA strands, both the single stranded DNAs, they cannot act as templates in order to organize the transcription. So here, if both the single stranded DNAs which proceeds in 3' to 5' polarity and as well as 5' to 3' polarity, if they act as templates, by that time, instead of the synthesis of single stranded mRNA, here double stranded mRNA, it successfully synthesizes. Is it clear? Instead of the synthesis of single stranded mRNA, double stranded mRNA, it successfully synthesizes if both the single stranded DNAs, if they act as templates. Is it clear? So let me arrange the things here. So here, as already mentioned, during the transcription mechanism, instead of taking the advantage of two template DNA strands, only one DNA strand which proceeds in 3' to 5' polarity, it is considered as template DNA in order to synthesize the RNA in 5' to 3' polarity. So here you make it a small clarification. Why both the DNA strands, they won't act as template in order to organize the transcription mechanism. So here if both the DNA strands, if they act as template in order to organize the transcription mechanism under such condition, instead of the synthesis of single-stranded mRNA, here double-stranded mRNA, it successfully synthesizes during the transcription mechanism. And in addition to this, once when the double-stranded mRNA, once when it participates in the translation mechanism, so here instead of the synthesis of one single polypeptide chain or protein, here two polypeptide chains or two proteins are synthesized. So here which is not at all acceptable and which is quite against to the central dogma of molecular biology. Is it clear? So let me write it on the board. In transcription, out of two DNA strands, only one DNA strand which proceeds in 3' to 5' polarity, 3' to 5' polarity act as template in order to synthesize RNA, in 5' to 3' polarity, 5' to 3' polarity. So here, in case of the transcription mechanism, why both the DNA strands, they won't act as template. So here, if both the DNA strands, if they act as template under such condition, instead of the synthesis of single-stranded RNA, here double-stranded RNA, it successfully synthesizes, which is quite against to the central dogma of molecular biology. Is it clear? So let me write it here. According to the central dogma of molecular biology, each and every gene, once when it undergoes expression during the transcription mechanism, it synthesizes a single-stranded mRNA. And here, the single-stranded mRNA, when it participates in the translation mechanism, it synthesizes one single polypeptide chain. It synthesizes one single polypeptide chain or protein. So here it is nothing but what? Central dogma of molecular biology. But here, if both the DNA strands, if they act as template in order to organize the transcription mechanism under such condition, instead of the synthesis of one single-stranded RNA, here double-stranded RNA, it successfully synthesizes during the transcription mechanism. And here, once when the double-stranded mRNA, when it participates in the translation, so here instead of the synthesis of one single polypeptide chain or protein, here two polypeptide chains or two proteins are synthesized, which is not at all acceptable and which is quite against to the central dogma of molecular biology. So the reason why during the transcription mechanism, instead of taking the advantage of two DNA strands as template, only one DNA strand, here it is considered as template in order to synthesize the RNA in 5' to 3' direction. Is it clear? Now here, in order to organize the transcription mechanism, there is a requirement of certain raw materials here. So here, what are the basic raw materials which are required for organizing the transcription mechanism? Here, the various raw materials which are required for organizing the transcription mechanism, they include, the very first one is RNTPs. Is it clear? So let me write it here. Raw materials which are required for organizing the transcription mechanism. The very first one is RNTPs. So here, what are RNTPs here? Ribose nucleoside triphosphates, and these include RATP, RUTP, RGTP, RCTP. Is it clear? So let me write it here. The various RNTPs, they include RATP, RUTP, RGTP and RCTP. So in case of replication mechanism, the specific nucleotides which are most commonly used in order to organize the replication, they are nothing but what? DNTPs, and they are nothing but what? Deoxynucleoside triphosphates. But whereas in case of the transcription mechanism, instead of taking the advantage of DNTPs, here RNTPs they are considered as the specific nucleotides in order to organize the transcription mechanism. And in addition to the RNTPs, there is a requirement of some more other enzymes, and these include DNA dependent RNA polymerase. Is it clear? So let me write it here. DNA dependent RNA polymerase. RNA polymerase. And the other one is sigma factor. Sigma factor. And the other one is rho factor. Rho factor. So here all these are the various raw materials which are required for organizing the transcription mechanism in case of prokaryotes. Is it clear? So here though the mechanism of transcription, it remains very similar in case of prokaryotes and even in case of eukaryotes, but still there are basic differences in between the prokaryotic transcription and even in case of the eukaryotic transcription. So here in case of prokaryotes, both transcription and as well as translation, they occur in cytoplasm. But whereas in case of eukaryotes, transcription mechanism, it occurs in nucleus, and whereas translation, it occurs in cytoplasm. Is it clear? So let me write it here. In prokaryotes, transcription and translation occurs in cytoplasm, because in case of prokaryotes there is no availability of well-defined nucleus or complex nucleus. So the reason why in case of prokaryotes both transcription and as well as translation, they occur in cytoplasm, but whereas in case of eukaryotes, so let me write it here. In eukaryotes, transcription occurs in nucleus, and whereas translation occurs in cytoplasm. And in addition to this, in prokaryotes, both transcription and as well as translation are coupled. So let me write it here. In prokaryotes, both transcription and translation are coupled. That means what does it mean? In case of prokaryotes, during the synthesis of mRNA, the specific mRNA or the nascent RNA, it successfully participates in the translation mechanism in order to synthesize the polypeptide chain. So the reason why in case of prokaryotes, both transcription and as well as translation are coupled. But whereas in case of eukaryotes, both transcription and as well as translation, they are not coupled. In case of eukaryotes, after the successful synthesis of eukaryotic mRNA in the nucleus, the specific eukaryotic mRNA, it has to export from the nucleus to the cytoplasm in order to participate in the translation mechanism or in order to participate in the protein synthesis. Is it clear? In case of prokaryotes, both transcription and as well as translation are coupled. But whereas in eukaryotes, eukaryotic mRNA which synthesizes in nucleus has to export to cytoplasm in order to synthesize polypeptide chain or protein. Is it clear? So here, in order to organize the transcription mechanism in case of prokaryotes, so here we are only concerned regarding the transcription mechanism which occurs only in case of prokaryotes but not regarding the eukaryotes, because in your NCERT textbook they have mentioned only the prokaryotic transcription but not regarding the eukaryotic transcription. So here we are only concerned regarding the prokaryotic transcription but not regarding the eukaryotic transcription. So here, in order to initiate the transcription mechanism in case of prokaryotes, there is a requirement of a specific enzyme which is nothing but what? DNA dependent RNA polymerase. So here the DNA dependent RNA polymerase enzyme, in order to initiate the transcription mechanism, it has to successfully bind to a specific region within the double stranded DNA which is nothing but what? Promoter. Is it clear? So let me erase the things here. So here, as already mentioned, in order to organize the transcription mechanism in case of prokaryotes, there is a requirement of a specific transcription unit. And here the specific transcription unit, it contains various components like promoter, structural gene and as well as terminator. So here the promoter and as well as the terminator, they flank the structural gene in case of the prokaryotic genomic sequence. And here most commonly the specific promoter, it relies or it remains at the upstream end in accordance with the polarity of sense strand or the coding strand. Is it clear? Most commonly the specific promoter, it relays or it remains at the upstream end in accordance with the polarity of sense strand or coding strand. And here likewise the promoter region, in order to inhibit the transcription mechanism, there is a requirement of one more other region within the double-stranded genomic sequence which is nothing but terminator. In what way? In order to initiate the transcription mechanism, there is a requirement of specific region within the double-stranded genomic sequence which is nothing but promoter. In the same way, in order to inhibit the transcription mechanism, there is a requirement of specific region within the double-stranded genomic sequence which is nothing but what? Terminator. Is it clear? So here the specific DNA strand which proceeds in 3' to 5' polarity, here it is termed as what? Template strand or antisense strand. Is it clear? So let me write it here. DNA strand which proceeds in 3' to 5' polarity, act as template in order to synthesize RNA in 5' to 3' polarity. 5' to 3' polarity. And the nascent RNA or the nascent mRNA which successfully synthesizes during the transcription mechanism, it remains very similar with the other strand which do not act as template, and here it is termed as what? Sense strand or the coding strand. Is it clear? The specific DNA strand which acts as template, it proceeds in 3' to 5' polarity, and the other strand which do not act as template in organizing the transcription mechanism, it proceeds in 5' to 3' polarity, and here it is termed as what? Sense strand or the coding strand. So here the nascent RNA or the nascent mRNA which successfully synthesizes during the transcription mechanism in 5' to 3' polarity, it remains very similar with the sense strand or the coding strand, except in case of the nascent mRNA there is an availability of uracil, but whereas in case of the specific sense strand or the coding strand, here there is an availability of thymine. So that is the basic difference. That is the only basic difference in between the sense strand or the coding strand and as well as the nascent mRNA. Is it clear? So let me write it here. Sense strand or coding strand proceeds in 5' to 3' polarity, and it remains very similar, with sense strand or coding strand, except in sense strand thymine is present, but whereas in nascent RNA which synthesizes during transcription, uracil is present. So that is the only basic difference in between the nascent mRNA which successfully synthesizes during the transcription mechanism and as well as the sense strand or the coding strand. Is it clear? So right now we will be studying regarding the mechanism and as well as the process which is involved in organizing the transcription mechanism in case of prokaryotes. Is it clear? So let me arrange the things here. So here, as already mentioned, in case of prokaryotes, in order to initiate the transcription mechanism, there is a requirement of a specific region within the double-stranded genomic sequence, which is nothing but promoter. So here, to the specific promoter region, an enzyme which is nothing but DNA-dependent RNA polymerase, it successfully binds. Binding of DNA-dependent RNA polymerase enzyme to the promoter region, it successfully leads to the initiation of transcription mechanism in case of prokaryotes. Is it clear? So let me write it here. DNA-dependent RNA polymerase binds to the promoter region. In order to initiate the transcription mechanism. And here, in between the promoter region and as well as the terminator region, here there is an availability of many more regulatory sequences which we will be studying during the regulation of gene expression. Is it clear? So let me write it here. In between promoter and terminator, many regulatory sequences are present. So here, in case of prokaryotes, during the transcription mechanism, 3 specific RNAs they are successfully synthesized, and these include rRNA, mRNA, and as well as tRNA. So here, by taking the advantage of one specific enzyme which is nothing but DNA-dependent RNA polymerase, all these 3 specific RNAs they are successfully synthesized during the transcription mechanism in case of prokaryotes. Is it clear? So let me write it here. In prokaryotes, 3 RNAs like rRNA, mRNA, and tRNA are synthesized by one DNA dependent RNA polymerase. By one DNA dependent RNA polymerase. So here these three RNAs like rRNA, mRNA and as well as tRNA, they play an important role in regulating the translation mechanism or the protein synthesis. So here the specific mRNA, it acts like template. So let me write it here. mRNA act like template during translation mechanism. And whereas tRNA act as adapter. tRNA, it acts as adapter in between the coding sequence of the mRNA and as well as the amino acids. Is it clear? tRNA acts as adapter in between the coding sequence of mRNA and amino acids. And in the same way, rRNA has catalytic activity. rRNA has catalytic activity, especially 23S rRNA. 23S rRNA in prokaryotes, and whereas 28S rRNA in eukaryotes. Is it clear? So here, in case of prokaryotes, by taking the advantage of one specific enzyme which is nothing but DNA dependent RNA polymerase, all these three RNAs like rRNA, mRNA and as well as tRNA, they are successfully synthesized during the transcription mechanism. Is it clear? So initially what exactly happens is, the specific DNA dependent RNA polymerase enzyme, it successfully binds to the promoter region. And here, the specific DNA dependent RNA polymerase enzyme itself, it is unable to recognize the promoter region. The DNA dependent RNA polymerase enzyme, in order to recognize the promoter region, it has to take the advantage of a specific factor which is nothing but what? Sigma factor. By taking the advantage of a specific factor known as sigma factor, the DNA dependent RNA polymerase enzyme, it is capable to recognize the promoter region. Is it clear? So let me arrange the things here. So here, as already mentioned in case of prokaryotes, in order to initiate the transcription mechanism, the DNA dependent RNA polymerase enzyme, it has to bind to the promoter region. But the DNA dependent RNA polymerase enzyme itself, it is unable to recognize the promoter. So here the DNA dependent RNA polymerase enzyme, in order to recognize the promoter, it takes the advantage of a specific factor which is nothing but what? Sigma factor. By taking the advantage of a specific factor which is nothing but sigma factor, DNA dependent RNA polymerase enzyme, it is capable to recognize the promoter region. Is it clear? So let me present it here. So here it is nothing but what? DNA dependent RNA polymerase. DNA dependent RNA polymerase. And here the DNA dependent RNA polymerase enzyme, in order to recognize the promoter region, it takes the advantage of a specific factor which is nothing but sigma factor. Sigma factor. So here by taking the advantage of sigma factor, DNA dependent RNA polymerase enzyme, it recognizes the promoter region. So here after recognizing the promoter region by the DNA dependent RNA polymerase enzyme, by taking the advantage of sigma factor, now here the DNA dependent RNA polymerase enzyme, it unwinds the double stranded genomic sequence. Is it clear? So let me present it here. The DNA dependent RNA polymerase enzyme, it is capable to unwind the double stranded genomic sequence. And here all these are nothing but what? The nucleotides, right? So here, according to the information which is present in case of the template DNA, the DNA-dependent RNA polymerase enzyme, it is capable to synthesize the nascent mRNA in 5' to 3' direction. Is it clear? So let me present it here. RNA polymerase, which is in association with sigma factor. It is capable to synthesize the nascent mRNA in 5' to 3' polarity. 5' to 3' polarity, right? 5' to 3' polarity. It is a nascent mRNA. Is successfully synthesized during the transcription mechanism by taking the advantage of DNA-dependent RNA polymerase enzyme. Is it clear? So here, these series of events they successfully continue until the DNA-dependent RNA polymerase enzyme it is about to reach the terminator region. So here, the DNA-dependent RNA polymerase enzyme, in what way, in order to recognize the specific promoter region, it takes the advantage of sigma factor. In the same way, the DNA-dependent RNA polymerase enzyme, in order to recognize the terminator region, it is capable to recognize the terminator region by taking the advantage of a specific factor, which is nothing but what? Rho factor. Is it clear? In what way, in order to initiate the promoter region, the DNA-dependent RNA polymerase enzyme, it takes the advantage of sigma factor. In the same way, the DNA-dependent RNA polymerase enzyme, in order to recognize the terminator region, it takes the advantage of a specific factor, which is nothing but what? Rho factor. Is it clear? So let me write it here. Rho factor is responsible for recognizing the terminator region. So here, DNA-dependent RNA polymerase enzyme, DNA-dependent RNA polymerase is supposed only to organize the elongation, right? The DNA-dependent RNA polymerase enzyme, it is capable to organize only the elongation phase of the transcription mechanism. But here, the initiation phase and as well as the termination phase of the transcription, in case of prokaryotes, they are successfully organized by taking the advantage of specific factors like sigma factor and as well as rho factor. Is it clear? So let me write it here. Initiation and termination of transcription are organized by taking the advantage of sigma factor and rho factor. So here, once when the DNA dependent RNA polymerase enzyme, once when it is about to reach the terminator region by taking the advantage of rho factor, here the nascent RNA successfully synthesizes during the transcription mechanism, it successfully dissociates from the double-stranded genomic sequence, and then after, even the RNA polymerase enzyme, it also successfully dissociates from the double-stranded genomic sequence. Is it clear? mRNA. So here it is nothing but what? The nascent mRNA. It successfully dissociates from the double-stranded genomic sequence, and even the DNA-dependent RNA polymerase enzyme, it also successfully dissociates from the double-stranded genomic sequence. Once when the RNA polymerase enzyme, once when it reaches the terminator region by taking the advantage of a specific factor known as rho factor. Is it clear? So here, though the transcription mechanism in case of prokaryotes, it remains very similar with the eukaryotic transcription, but still there are basic differences in between the prokaryotic transcription and as well as eukaryotic transcription. So right now we will be studying regarding the basic differences in between the prokaryotic transcription and as well as eukaryotic transcription. Is it clear? So let me erase the things here. So here, as I have already mentioned you, the prokaryotic transcription mechanism, it remains vary from the eukaryotic transcription mechanism in certain aspects. So here, in which particular aspects the prokaryotic transcription mechanism, it remains vary from the eukaryotic transcription mechanism. Here, in case of the prokaryotic transcription mechanism, by taking the advantage of one single DNA dependent RNA polymerase, all the various RNAs, they are successfully synthesized. But whereas in case of the eukaryotic transcription mechanism, in order to synthesize the various RNAs, there is a requirement of various RNA polymerases. Is it clear? So let me write it here. In prokaryotic transcription mechanism, in prokaryotic transcription mechanism, only one DNA dependent RNA polymerase is used to synthesize various RNAs. But in eukaryotic transcription, in order to synthesize various RNAs, there is a requirement of various RNA polymerases. Is it clear? In order to organize transcription, there is a requirement of various RNA polymerases. And these include RNA polymerase 1, So here RNA polymerase 1, it is the enzyme which is responsible for the biosynthesis of 5.8S rRNA, 18S rRNA and as well as 28S rRNA. Is it clear? 5.8S rRNA, 18S rRNA and 28S rRNA. And whereas RNA polymerase 2, it is responsible for the biosynthesis of hnRNA or mRNA. Then in the same way, RNA polymerase 3, it is responsible for the biosynthesis of 23S rRNA, 5S rRNA and tRNA. Is it clear? And in addition to this, in case of prokaryotic transcription mechanism and as well as eukaryotic transcription mechanism, there are certain other differences here. So here these include, in case of prokaryotes, both transcription and as well as translation, they are coupled. Is it clear? In case of prokaryotes, so let me write it here. In prokaryotes, both transcription and translation are coupled. But whereas in eukaryotes, transcription occurs in nucleus and translation in cytoplasm. And in addition to this, in case of the eukaryotic transcription mechanism, after the successful synthesis of eukaryotic mRNA during the transcription mechanism, the eukaryotic mRNA, it has to export from the nucleus to the cytoplasm in order to participate in the translation. Is it clear? So let me show it here. Let us assume it has the nucleus here. Nucleus. And here it is nothing but what? Cytoplasm. So here, in case of the nucleus, during the transcription mechanism, mRNA successfully synthesizes here. So here, prior to the exporting of the eukaryotic mRNA from the nucleus to the cytoplasm, the eukaryotic mRNA, it undergoes specific modifications, and these include post-transcriptional modifications. Is it clear? So let me write it here. Prior to the export of eukaryotic mRNA from nucleus to cytoplasm, eukaryotic mRNA undergoes post-transcriptional modifications. Is it clear? So here, the various post-transcriptional modifications which most commonly occurs in case of the eukaryotic transcription mechanism, these include 5' capping and the other one is 3' tailing. Is it clear? So let me write it here. 5' capping and the other one is 3' tailing. So here what exactly happens in case of 5' capping here? In case of the 5' capping, successful recruitment of 7-methylated guanosine nucleotide at the extreme 5' end of the eukaryotic mRNA takes place. Is it clear? 7-methylated guanosine nucleotide recruits at 5' end of eukaryotic mRNA. And here, in case of the 3' tailing, successful recruitment of multiple adenosine nucleotides at the extreme 3' end of the eukaryotic mRNA takes place. And here, for the successful recruitment of multiple adenosine nucleotides at the extreme 3' end of the eukaryotic mRNA, the specific mechanism, it takes the advantage of an enzyme which is nothing but what? Polymerase, which is a template independent enzyme. Is it clear? So let me write it here. Multiple adenosine nucleotides are added at extreme 3' end of eukaryotic mRNA by polymerase, which is a template independent enzyme. So here nearly 250 to 300 adenosine residues are added here. 250 to 300 adenosine residues, they recruit at the extreme 3' end of the eukaryotic mRNA. At the extreme 5' end of the eukaryotic mRNA, 7-methylated guanosine nucleotide, it successfully recruits here. And here it is termed as what? 5' capping. and here in case of the 3' tailing, multiple adenosine residues around 250 to 300, they successfully recruit at the extreme 3' end of the eukaryotic mRNA by taking the advantage of a specific enzyme which is nothing but what? Polymerase, which is a template independent enzyme. So let me write it here. Template independent enzyme. Is it clear? So here these are nothing but what? Post-transcriptional modifications. So once when the post-transcriptional modifications, once when they are successfully organized in case of the eukaryotic mRNA, now the specific eukaryotic mRNA, it is ready to export from the nucleus to the cytoplasm in order to participate in the translation mechanism. Is it clear? And in addition to this, in case of the eukaryotic mRNA, so let me write it here. In case of eukaryotic mRNA, in addition with coding sequences, which are otherwise known as exons, there are non-coding sequences. Which are otherwise known as introns. So here these non-coding sequences are introns. They are successfully spliced out from the primary transcript mRNA. So here once when the non-coding sequences are introns, once when they are successfully spliced out from the primary transcript, now here from the primary transcript, secondary transcript or mature RNA, it successfully produces here. Is it clear? So let me arrange the things here. So here, in addition to the post-transcriptional modifications like 5' capping and as well as 3' tailing, one more other processing event, even it also occurs in case of the eukaryotic mRNA, which is nothing but what? RNA splicing. So here in case of RNA splicing, what exactly happens is, the non-coding sequences are the introns which are present. In case of the primary transcript mRNA, they are successfully spliced out. So here once when the non-coding sequences are introns, once when they are successfully spliced out from the primary transcript, secondary transcript or mature RNA or intron-free RNA, it successfully synthesizes here. Only the intron-free RNA or mature RNA, it is capable to participate in case of the translation mechanism in order to synthesize the polypeptide chain. Is it clear? So let me present it here. So here it is nothing but what? The primary transcript. Primary transcript, right? Primary transcript in the sense the specific mRNA or the nascent mRNA is successfully synthesized during the transcription mechanism in case of eukaryotes here. Primary transcript. So here the primary transcript, in addition with the coding sequences known as exons, it also contains the non-coding sequences known as introns. So let me present it here. Exon. So here it is exon and again it is intron. Exon, intron, intron, exon. So here the coding sequences are termed as exons. So let me write it here. Coding sequences are termed as exons, and whereas non-coding sequences are termed as introns. So here the non-coding sequences are introns. They undergo successful splicing by taking the advantage of a specific processing mechanism which is nothing but what? RNA splicing. Is it clear? So here during the RNA splicing mechanism, the non-coding sequences are introns, they are successfully spliced out here. So here once when the non-coding sequences are introns, once when they are successfully spliced out from the primary transcript, now from the primary transcript, secondary transcript or mature RNA or intron-free RNA, it successfully synthesizes here. So here it is nothing but what? Intron-free RNA. intron-free RNA or mature RNA or secondary transcript. Is it clear? Only the specific secondary transcript or mature RNA or intron-free RNA, it is capable to export from the nucleus to the cytoplasm in order to participate in the translation mechanism in order to synthesize the polypeptide chain or proteins. Is it clear? So initially many of the biologists, they have assumed that only DNA is having the biological importance but not RNA. Is it clear? Because the specific DNA only contains the coding sequences. But in recent times, many of the biologists, they came to know that here not only DNA, even the particular RNA, it is also having the biological importance, because the specific RNA which successfully synthesized during the transcription mechanism in case of eukaryotes, it not only contains the coding sequences known as exons, and even it also contains the non-coding sequences known as introns. So here the specific non-coding sequences known as introns, they are successfully spliced out by taking the advantage of a specific mechanism known as what? RNA splicing. So here why the specific non-coding sequences are introns, they are successfully spliced out here? Because the specific eukaryotic mRNA, it is also having the biological importance. So the reason why the specific non-coding sequences are introns, they are successfully spliced out from the eukaryotic mRNA prior to the exporting of the eukaryotic mRNA from the nucleus to the cytoplasm in order to participate in the translation mechanism. Is it clear? So here this is regarding the mechanism of transcription from the molecular basis of inheritance in detail. And in the next upcoming lecture session, we will be studying regarding the translation mechanism in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.